Okay, finally we're going to go to a yak vector. And yak vectors are really weird. Now the great thing about the yip vectors I wanted to say is they're so much more stable than the 2 micrometer plasmids, because the 2 micrometer plasmids can be lost. Once you've integrated into the yeast genome, you're there, and you'll always be there. So if you want to use yeast for example, in the human body, or you wanted it as a vehicle, or you wanted to use them in a big bioreactor vat, something like that, where they're going to be multiplying hundreds of times, probably be better to use a yip vector than a 2 micrometer. If you just want to do quick experiments, it's better to use the 2 micrometer. Okay, let's look at yak vectors. Yaks are really weird, so I'll just tell you that right now. So a yak vector is going to be a yeast artificial chromosome, a yeast artificial chromosome. So with this, we have the blue triangle is a telomere sequence. And then this dot right here is a centromere sequence. And then the brown part is the ORI sequence, the autonomously replicating sequence from yeast. So we have an ORI, we've got a centromere, and we've got 2 telomeres. That's a chromosome. Just that's the smallest definition of a chromosome. So what you're going to do is you're going to take this vector here, and then you're going to cut it with BAM-H1. And in between the 2 telomeres, we're going to have in between the 2 telomeres, we're just going to cut that. That's a stuffer fragment, And then you're going to have this separated, and then you also cut it with echo R1, which is right here. And now we have left arm of the chromosome, right arm of the chromosome, and into this we put our, let's say we have an entire genome which has been cut up with echo R1, and we put these fragments into this slot, and now we have a library, a yak library, and we have all these chromosomes. And then we transform yeast with these chromosomes, and then each yeast gets an extra chromosome, which contains a very large piece of DNA. And so that's basically how yaks work. Yaks are most typically done in mapping of the chromosome. All right, next we're going to go to baculovirus expression. So we've been making protein. Now we're going to go to baculovirus expression, and let's see what page I wanted to go to here. Actually, the one I do want to go first of all to Wikipedia, because it had a really nice section on baculoviruses. Okay, first of all, what are baculoviruses? Let's look at one of them here. There are 2 different genera in baculoviruses, so even viruses can be divided up into genera. These are insect viruses, and you're thinking, why are we studying insect viruses to study gene expression? But you'll see, one gene is the NPVs, the other is the GVs, and so we'll go to the NPVs, the nucleopolyhedrosis viruses. And here they are. They infect caterpillars, and look at this caterpillar here. It says, another victim of nuclear polyhedrosis virus. So that's something you should see on the New York Times. But this is really, truly ghastly. If you look at that caterpillar there, it is hanging at the end of a twig. And so, like rabies, this virus actually causes a certain behavior. And so what it does is it lives in the soil. It can live into the soil for over a decade. this virus. It is impervious to UV light. It is impervious to microbial degradation, as you will see. And it can live for a long time. When it gets into the insects eat this, and then they will undergo just like Ebola, all of the compartments will break up, all the compartments will actually liquefy, and the insect will be totally liquefied inside of its exoskeleton, which is really bizarre. And I worked with these for a while, and I went down to Texas A&M and I picked some up. And the guy inoculated the caterpillars for me, and I brought it back to Waco, and he said in about 3 to 4 days, you should have a completion of the cycle. And I said, oh, really? And so I was looking forward to this. But after 4 days, the caterpillars were still walking around. And I said, well, that's strange. They're kind of walking slowly. And I said, well, what's going on there? And so I took a little probe, and I touched the caterpillar just to see what's going on. And it burst like a balloon.
It burst.
It burst. And so it was walking, but then it just went floof, and then there's nothing left but this bag and all the guts everywhere. And I just barely touched it. And I said, how in the world were you walking 2 seconds ago? So it is like Ebola on a grand scale. It's a horrible death. And what this thing, and so this is a sac of virus, basically, hanging on the edge of a twig. And what it does is at the last gasp of breath, well, they don't breathe, but anyway, the last bit of muscle motion that the insects have, they go to the end of a twig, and there they die, and then they form this case, and then they dry up, and then the virus goes everywhere. And that's how it spreads. So if you think we have bad viruses, this is way worse than anything we have. If you can imagine something 20 times worse than Ebola, yet stays in the soil for 10 years. And a lot of people, some insect ecologists believe that this actually is what keeps insects in check, so they don't completely overpower the earth. It might be this virus is actually keeping everything in check. So it might be much more important than you think. But this is why it's so resistant. You see these things in here? Those are the actual virions. Those are the actual viral particles. All this other stuff, it's protein. So it's polyhedron protein. And these things are totally occluded, totally encapsulated. And that's why they can survive in the broad daylight, in UV, or in soil, in a very hostile microbial environment. So someone said, that's just an incredible virus. And then someone was really smart and said, how did it make all that protein? That's a good question, isn't it? And the reason it made all that protein is that it is just unbelievably, has an unbelievable promoter. Unfortunately, the promoter only works in insect cells. That's really bad, isn't it? And insect cells are able to work, insect cells are as difficult to work with as mammalian cells. So that's your big drawback. You can make gobs of protein with this, but you have to use insect cells. Second drawback is that this virus is huge. It's 130 kb, which puts it almost in the league with vaccinia virus. I mean, it's huge, herpes viruses. So, two things, gigantic DNA that can't be made smaller, and also it has, well, it can be made somewhat smaller, and also has to be done in insect cells. So, this is, how do you work with something like this? You do it by, you do it with a system which is called a BACMID system. And let me see if I can give you a picture of a BACMID. Okay, I think this one actually will do it. Yeah, here we go. Okay, so with a BACMID, what you do is you do all your work in E. coli, just the way you always do. And then you, so here is your donor plasmid, and you work with your GOI, gene of interest, in your donor plasmid. Then you take this donor plasmid and transform it into E. coli, which contains a BACMID. And BACMID is very, very large. What a BACMID is, it's a shuttle vector that allows you to go from E. coli into insect cells. That's what a BACMID is. It's a shuttle vector between E. coli and insect cells. It has origins of replication and all that kind of stuff for E. coli as well as insect cells. So it's so big that you have to keep it in a separate cell. So you could buy these DH10, which is a type of E. coli, BAC cells, DH10 BAC cells, competent cells. So you take these competent cells, you add the donor plasmid here, it recombines by it's recombination, homologous recombination, and then now it's in here, and then you take and you extract the backman DNA, and then you go ahead and put it into insect cells, and you get these virus particles. So that's basically what you have to do. If you want to use the baculovirus system, then that's what you have to do. Okay, let's see if it's working. Okay, great. All right, let's go on then to mammalian cell expression. And this is not as difficult as you would think, the mammalian cell expression. We're just going to look at two of them. Mammalian expression vector, we'll take that one. And if you look at these, they're pretty straight up. Let's see, here's one. So here we have a plasmid. It's got an ampicillin resistance in it, right? And and it's also got a PBR322 origin. That is a classic plasmid origin for replication in bacteria. And it's got a F1 origin, which is going to allow you to make single-stranded DNA if you wanted to sequence it or make probes. So it's got all the stuff you need for growing in E. coli. But it also has this SV40 origin, and that origin is SV40. Does anyone know what SV40 is? It's simian virus 40. So it is a eukaryotic virus. So it has an origin that allows replication in mammalian cells. And so this is really a shuttle vector. And then here's what you're going to see in almost all of these mammalian cell expression vectors, a CMV promoter. And CMV is another virus, and you notice we're using viruses all the time because they have powerful promoters. CMV is cytomegalovirus. Cytomegalovirus. So CMV is cytomegalovirus, and that is the promoter that you see almost all the time in these mammalian cell vectors. And then we have a poly-A right here, and we have various poly-A's. And that, what they mean by a poly-A is not a string of A's, but it is a poly-A signal. So it is a specialized signal. It's the transcriptional stop signal. And so the upstream for that is AAA, AAU, AAA. But there's a downstream signal and so forth. But this tells the transcriptional machinery of the mammalian cell, hey, we're stopping transcription here. And furthermore, we're going to add a poly-A site here. We're going to add a string of A's. So that's what they call a poly-A. Transcriptional stop, and this is where we're going to add our A's. Okay? So that's basically it. And if you look at all the other ones, they look pretty much the same. They have a few other tricks to them, but see, here's a CMV promoter, and then here we have an intron, which actually helps in expression, but here we have an SV40. Now we have an SV40 from, a poly-A from SV40 that we're borrowing, but we have ampicillin resistance and an ORI for bacteria and so forth. So if you look at all these, they're kind of the same. Now, the difference, one difference we could have is you can also have lentivirus. And lentivirus expression vectors are going to be a little different. So let's look at this one right here.